Okay, so we are moving on to topic 3. I'm going to start with 3.1, which deals with enzymes, and more particularly enzyme structure. I'll try to be as basic as I can while maintaining some level of depth. Enzymes, if you're looking down here, this is something you might have seen before. My cursor here we are. Down here in the bottom right corner is a protein. That's something you should be able to recognize due to the fact that this is the tertiary structure of a protein. This is how it folds. You can see the evidence of the secondary structure in here as you look at these alpha coils and these beta sheets, which have arrows on them indicating the directionality of this protein. But enzymes typically are proteins. They are biological catalysts. This word catalyst is something you should remember from chemistry, but these speed up chemical reactions. These speed up chemical reactions. Typically, enzymes are proteins. Sometimes, though, they are big consist of RNA. If you remember when we talked a while back about the RNA world hypothesis, we talked about RNA and how it can form complex structures, and they can do some of these things that enzymes can do. Sometimes the RNA, these help to facilitate most chemical reactions in your body have an enzyme involved at some level. This includes the replication of DNA and even the creation of proteins in the first place at the ribosome. So there's a lot of proteins involved in every aspect of this. They're required for most biological reactions. They are highly specific. We'll get more into that in a later video. But one enzyme typically is one enzyme per one substrate, typically. These control every reaction of life. They are part of almost every metabolic reaction in our body. You don't get too far in biology without having to recognize the power of these enzymes and flexibility of these enzymes. So what we're looking here right now is an enzyme and substrate interacting. That's what this picture is. First word I need you to know when we talk about enzymes is substrate. If you remember that example I may have done in class, the H2O2 reaction, right? That H2O2 would have been our substrate, right? Our enzyme for that reaction was an enzyme called catalase, which I kind of generated with that starch that activated, or the yeast rather. Activated yeast generated this catalase, which is able to break down this H2O. But the substrate in this reaction, or the thing that's going to be on one side of the formula right here, right? H2O2. Substrate is what enters the active site. This is what binds to the enzyme. The enzyme substrate complex, which is ES complex, is this right here. What this means is, and I'm going to erase this real quick. I'll draw this formula again when I have some more room over here. Enzyme substrate complex is the temporary association of enzyme with the substrate. Substrate enters, enzyme, it's bonded to the enzyme. This is the enzyme substrate complex. The products, and in our specific example, of course, we had O2 as one of our products, and H2O is another product in this reaction, right? This was for our H2O2 example here. This is the end result, right? Catalase was breaking down this H2O2. The active site simply refers to this site where the substrate will enter. This is considered the catalytic site. You should see that word. You'll see this word catalysis again. Substrate fits into the active site. The enzyme breaks down this substrate or builds up the combination of substrates into a single product. But these are the three words that are hugely important for you to know. Substrate, product, active site, and enzyme, of course. If we were to write this formula down for the reaction we did in class, H2O2, our substrate was being broken down into, to H2O and O2, right? And typically the enzyme is written above. You'll also notice with enzymes, I'm not going to talk about too many other specific ones here, but they typically end in ASE. I talked about a few more in class already, but typically these end in ASE when we're talking about enzymes. A lot of them do anyways, not all of them, but a lot of them end in ASE. That's one way you know you're looking at an enzyme. Okay, The lock and key model is often the way these enzymes are perceived. This is one way of looking at enzymes, being that you have a substrate, which is very specific. This is like the key. The substrate is like the key, and the enzyme is like the lock, right? You have a substrate enter. It causes some sort of reaction which unlocks this enzyme. This obviously isn't the best example because there is no locking mechanism on these. So an induced fit model is a better way of looking at this. And the way this works is you have a substrate which, as it enters, causes a conformational change in the enzyme, meaning this enzyme typically changes shape to fit the substrate and in order to allow this reaction to proceed. So conformational simply refers to a change in shape of an organic molecule, and this is what this induced fit model says. Now, it is important to remember that these enzymes are highly specific, meaning that, and this is the reason this lock and key model is a good way to look at it, a certain substrate fits into a very specific enzyme. In other words, one enzyme for every substrate, right? One enzyme, one substrate. Highly specific. This lock and key model kind of illustrates that pretty well, I think. Okay, that's it for this video. Thank you for listening, and I will see you in 3.2.